Hi there and welcome to this A Level Biology revision video, Mr. Phillips, and today we're looking at translocation even in the plant transport module, as long as I can get my words out. So let's have a look then. So first of all, we need to know where is translocation happening, and this is obviously happening in the phloem, and the phloem is a tissue. So remember, quite important basic thing here, tissues are groups of cells that work together for the same function. So in this case, there are actually two groups of cells, which a lot of people overlook. So the first type of cell is the sieve tube element, okay, and that in this case is these cells here, okay, and then we have the companion cells, which are these cells here. So the sieve tube element is the cells that the assimilates of photosynthesis, the sucrose most of the time moves up or down through. And they are very simplified cells in they've got reduced cytoplasm. So they have no nucleus, they have very little cytoplasm because they have been adapted to allow the translocation through them. They end up line up end to end to make a tube, and they contain cross walls. So if you see this bit here, okay, at intervals, but they have pores in them to allow the movement of material up and from one cell to the next, and those pores are called sieve plates. They are very thin tubes, and they're normally a hexagonal sort of shape. Next bit then is the companion cells. So the companion cells, I always say, is like a life support system for the sieve tube elements. They are small, but they've got a very large nucleus for their size, okay, and they've got a dense cytoplasm, meaning they've got a lot of mitochondria, a lot of ribosomes, because effectively they're keeping the sieve tube elements alive. So they need like double the amount of content. So they also have a high mitochondria because they need a lot of ATP for active transport, which we're going to be talking about in a moment. And they carry out metabolic processes, I said, for the sieve tube elements. And they're linked through gaps in the cell wall called plasmodesmata. But you have to, so that is this bit here, if you can't see, okay, but you have to be aware that these plasmodesmata, so they're there, is actually a continuation of the cytoplasm. So all this cytoplasm in here continues into the sieve tube elements. And there's a picture just come up of the sieve tube plates to show you where the materials be going. So what is translocation? Well, translocation is the movement of organic substances, which is sugars, mainly sucrose, and other chemicals made by the plant. And it happens in the phloem as sucrose. And the key thing for phloem, I think, which we need to mention before we say anything else, is that it can go up and down in the case of translocation. It always goes from the source, which is the place where the sucrose is loaded into the phloem, okay, to the sink where it is removed from the phloem. So the source might be at the leaf, and the sink might be at the flower, or the sink might be at the roots, okay, but that is where it goes from. So how does this process work? Well, if we start off, the first thing happens is sucrose, the whole process is that sucrose is going to be actively loaded into the phloem. So how does this work? So if we cross off number 1 for a minute and we talk about number 2. So number 2, the hydrogen ions are actively pumped out of the companion cells into the surrounding cells. So imagine this is your leaf and there's lots of cells here, okay, and what we're saying is hydrogen ions are pumped out of this cell, so we end up with lots and lots of hydrogen ions in these cells here. So we end up having a concentration gradient, lots of hydrogen ions, and these hydrogen ions want to diffuse back into the companion cell. However, also in those cells there is sucrose. Now sucrose is not represented with the letter S, obviously, because sucrose is a complex structure, okay, but for the minute let's imagine it is just the letter S. And basically for one of those hydrogen cells to come back, it has to come through a co-transporter protein, okay, and that co-transporter protein will only allow hydrogen ions to come through if they bring sucrose with it. So the hydrogen ions want to move from a high concentration to a low concentration back into the companion cells, but they can only do it through a co-transporter protein, and they have to bring sucrose with them in order to do that. We then get a buildup of the sucrose. So the sucrose builds up in the companion cells as a result, meaning it's got a high concentration. So it then moves from a high concentration to a low concentration through the plasmodesmata into the xylem at the source. Okay, that's the point where the sucrose is loaded into the xylem. So that means then we have a high concentration of sucrose. So then why does this cause movement? Even though you're building it up, why does that automatically cause movement? So as we just said, in that process that we just described, we end up with lots and lots of sucrose in the by the source of the phloem cells. That then reduces the water potential. Okay, so the symbol for water potential is a W with a stick underneath it. Not that I've drawn that very well. I'll have to redo that. I can't cope with that poorness. There we go. Okay, so there we go. And basically what happens is water in the surrounding cells, and don't forget the xylem are right next to the phloem in the vascular bundle, water in the surrounding cells is then drawn in due to osmosis from a high water potential to a low water potential. This creates what we call a high hydrostatic pressure. So the pressure here at this end, at the source end, is extremely high because you've just pumped all this water in, okay, and then the water then moves away from this, either up or down the phloem, okay, until it gets to where the materials are going to come out. Now, what happens is here, the water moves down, okay, and the sucrose gets removed. Oops, sorry, I've still got the wrong bit on here. The sucrose gets removed from the into the cells that need it and require it, and we actively transport it to where it's needed. Because the sucrose then leaves, this means that these cells here have got a low water potential, and the water follows out via osmosis. That creates a pressure difference between the source and the sink. So the source of the loading of the phloem has a high pressure, the sink has a low pressure. So this causes mass transport movement from high to low pressure. So let's summarize that then. The source, the sucrose, is actively loaded. Water follows it into the phloem via osmosis from the xylem, raising the hydrostatic pressure. At the sink, the sucrose is used in cells for respiration or stored as starch, so it moves from the phloem into the cells that need it via active transport and diffusion. And then the water potential becomes higher in the sieve tube elements as the sugar leaves, so the water follows it out via osmosis, lowering the pressure. So this pressure difference is what causes a gradient between the high pressure to the low pressure, and it's due to the fact that water moves in and out from the xylem due to changes in water potential at the source and the sink. So what evidence do we actually have for this? What evidence do we have that this is how sugar is moving? Well, one piece of evidence that we have is using radioactively labelled carbon dioxide, doing something called an autoradiograph. And basically what you could do is you could radioactively label carbon dioxide, and when that carbon dioxide is then assimilated into glucose, that gets converted into sucrose, and you can time, and over time you can see that carbon dioxide travelling through the phloem, showing you that this happens. Also, if we puncture the phloem, then the sugar will actually, or the sap which contains sucrose, will actually ooze out, which shown us as a high pressure near the source. And phloem sample near the sink also has a higher sucrose concentration, I should say, sorry, near the source rather than the sink. And there's also evidence of mass flow, because if we look at the content of the sucrose levels in the leaves compared to the sucrose levels in the phloem, they will effectively follow each other. So when the leaves have got loads of sucrose, it will then be followed into. Viruses put in leaves will also generally move down the phloem in light, showing you the movement away from the leaves. And when they're putting the leaves in the dark, they'll stop in the leaves, and therefore, again, it shows nighttime mass flow isn't happening. And you can also do ringing of trees because the phloem are known to be on the outside of the stem, and if you just cut the outside of the stem off, you get a sink of sugar collecting there, showing you that it's moved down. The below bit doesn't bulge, showing you that mainly it's moving down. And again, you can look at this. This shows you carbon dioxide at different times, and you can see carbon dioxide can be monitored moving around the plant to show the movement in the phloem. And that's obviously not carbon dioxide, it's sucrose, which has got carbon radioactively labeled in it.